Okay, so in this presentation, we're going to go over the nervous system, but more in particular, we're going to study neurons and how electrical signals are transmitted from cell to cell. So let's get started. So when we just do a quick overview on the nervous system, the nervous system is like any other organ system. It's made from a collection of organs, which are made from a collection of tissues, which are made from a collection of cells. So the nervous system is a network of these. What's unique about the nervous system is that it controls thoughts, movements, and various life processes. And how it does that, how the nervous system does this, is through sending of signals, electrical signals to and from the brain. And that's kind of what we're going to look at in a little bit. The nervous system responds very quickly. You can try this at home. If you just turn all the lights on, you and a friend turn all the lights on and get real close to one another and look into each other's eyes. And then on the count of three, turn the lights off. And when you do, you'll notice that almost immediately the pupil gets bigger. Well, that's because, again, the dark light causes nerves in your eyes to readjust. And so the point is that the nervous system responds to change very, very quickly. So you think about, well, what must happen in order for this person to pick up the apple that's sitting on this table? Well, it's kind of neat. Light has to first reflect off of the apple. You saw the white arrow enter and the red arrow reflect. White light enters, red reflects to the person's eyes. Well, your eyes contain photoreceptors called rods and cone cells that will send an electrical signal to your brain. Your brain will then receive the signal and send a message, an electrical signal, down your spine, down your arm. Eventually, that signal will reach your hand. Now, the muscles in your hand will react to that electrical signal. Watch what happens. You start reaching for the apple, and eventually you grab the apple and you can consume it. But it's a lot of actions of your nervous system that goes into just picking up an object. So when we look at neurons, what neurons are another name for nerve cells. And so here's kind of a typical drawing of a nerve cell here. The function of a nerve cell is to transfer electrical signals, and we call these signals impulses. So a neuron is going to send impulses, electrical signals, to the brain and from the brain. So there's three main parts to a neuron. I just highlighted the cell body. You might also hear this called the soma, S-O-M-A, soma. And the cell body or the soma is what contains the nucleus and all the organelles that are fairly typical to a eukaryotic cell. Notice there's little branches coming off of the cell body. These little branches are called dendrites. And what they're going to do is they're going to receive a signal from a neighboring cell. In my animation, there's an impulse. That spiked flashing yellow object is an impulse, and it's coming down from the neuron on top. Well, a dendrite in the middle neuron just received an impulse, and eventually that impulse is going to travel down that long straightaway called an axon. So in my animation, the impulse has just traveled down the axon and has reached the end of the axon. And so what happens next? Well, we'll get into that in just a few moments. Before we get into how neurons actually send signals, let's look at the three different types of neurons. The first are what are called sensory neurons. And these are the neurons that detect our senses. They detect stimuli and they transmit signals to and from the brain. So in our little animation here, let's say that this lady has her hand over a very tall candle, but there are sensory neurons in her hand that will sense heat and will send a signal to her brain saying to pull your hand away. So your sensory neurons are related to your five senses, your sense of sight, your sense of smell, your sense of hearing, your sense of taste, your sense of touch. In your nose, you have sensory neurons to detect smell. On your tongue, you have sensory neurons to detect taste. The second type of neuron are what are called interneurons. And the reason for the picture is because these are located inside of the brain. And the purpose of the interneurons are to receive signals, to receive the messages from your sensory neurons. So a moment ago we saw that cartoon lady touching a hot candle. Well, the sensory neurons in that woman's hand sent a signal up to her brain. So the sensory neurons in that woman's hand sent a signal to the interneurons in that woman's brain. Eventually she'll pull away from the hot candle. And that brings us to the third type of neuron called motor neurons. And what is going to cause her to pull away from a hot candle if you're touching a hot candle? Well, that's going to be the job of your motor neurons. So in that example from a moment ago with the lady touching the hot candle, her brain will send a signal back to her hand, and that's what we see here. We see a signal going back to the woman's hand, and in our hand are muscles. So the motor neuron is going to pass signals to other tissue. In this case, the motor neuron is sending a signal to the muscles in that woman's hand who was just a moment ago touching the hot candle. And so we can see, look at the far right of the motor neuron. You can see that the motor neuron is actually connected to the muscle cells. And when the muscle cells receive that electrical signal, the muscle cells will stretch and contract. Basically, the woman will pull away from the heat. So these are the three types of neurons here. Well, now let's focus on how a neuron actually sends a signal. So there's a state I want to describe when a neuron is at rest. A neuron has what's called resting potential. When it's at rest, it's not in the process of sending a message, not in the process of sending an electrical signal. And so let's zoom on into this neuron here. Let's look at the cell membrane of this neuron. Okay, so we can see that the top of the picture is the exterior of the neuron and the bottom of the picture is the interior of the neuron. Well, we can actually insert an electrode, a little tiny micro electrode into the neuron to measure the strength of the electrical charge. And by doing this, we've actually discovered that the neuron's interior has a negative charge because there's a lot of proteins that are inside of the cytoplasm. So because of all those negatively charged proteins that just popped up, the inside of a neuron typically has a negative charge. Now, scattered amongst the negative proteins are a few positive potassium ions. However, they're just not in as large numbers as all the negative potassiums. So because of the negative proteins. So because of all those negative proteins, the internal charge is around negative 70 millivolts. And when we apply the electrode to the outside of the neuron, we discover that the exterior, the outside, has a positive charge. Well, that's because there's an abundance of sodium ions. And so that causes the exterior, the outside of the neuron, to have a positive charge. So when we look at the full picture, here's what we see. Exterior positive charge because of the sodiums, interior negative charge because of all the proteins. However, there are a few positive potassiums scattered about. And those positive potassiums are actually going to be really important. So now I want to discuss an action potential when a neuron is actually sending a signal. And an analogy I like to make is that an action potential is kind of like people at a sporting event doing the wave. So here's people doing the wave at a sporting event. One person stands up and then sits down, and then the process repeats. And so an action potential is kind of like the people doing the wave at a sporting event. And I hope you'll understand the analogy once we go through it. So let's actually show you now how a neuron generates an impulse. In the picture, we see sodium, positively charged sodium on the outside, positively charged potassium on the inside. I just did not draw the negatively charged proteins in this picture because I didn't want to include too much clutter in the picture. But please remember, the inside of a cell, the inside of a neuron, has a negative charge. So when an impulse occurs, around negative 40 millivolts, that black set of doors on the left, that's a sodium channel. That black sodium channel is going to open. And when that happens, the positive sodium is going to diffuse from a high concentration to a low and go into the cell. That influx of sodium is going to bring the charge from negative 40 all the way up to positive 50 millivolts. That's the impulse. An impulse is that change from negative to positive charge. That's what the impulse is. And so what happens next? Now that the internal charge is positive 50, well, after that happens, around positive 50, notice how the black sodium doors on the left close, but around positive 50, the red doors, the red potassium doors are going to open. The potassium channels are going to open, and that's going to allow potassium to diffuse from a high concentration and exit the cell. That's going to drop the charge all the way down to negative 90. So the charge went from negative to positive back to negative. At negative 90, that red doorway, that red set of doors, that red potassium channel is going to close, and now again the area inside is back to being a negative charge. This process is simply going to continue down the axon, down the length of the neuron. Let's take a closer look at this. So let's look at this in a little more detail. Here's a couple neurons in close proximity to each other, and I'm going to have the neuron on the left send an impulse down its axon. Well, let's freeze the impulse and let's actually zoom in for a closer look. And when we do, we see a bunch of positive sodium on the outside, a bunch of positive potassium on the inside. It might look like the inside is positive because of all the potassium. Remind yourself, the inside actually has a negative charge because of a lot of negative proteins that are inside the cell. I just didn't draw the negative proteins because I didn't want to put too much clutter. But remind yourself, the internal charge is negative. So when we now look at an action potential, at negative 40 millivolts, the black sodium doors are going to open. That's going to cause a bunch of sodium to diffuse into the cell, bringing the charge from negative 40 all the way up to positive 50. At positive 50, those black sodium channels are going to respond and close. But by the way, that positive 50 right there, that's the impulse. An impulse is that change from negative 40 to positive 50. So at positive 50, like I just said, those black sodium channels are going to close, and the next set of doors on the right, that red set of doors on the right, those are potassium channels. At positive 50, the potassium channels are going to open, and then a bunch of potassium is going to exit and diffuse out of the cell. That's going to drop the charge from positive 50 all the way down to negative 90. And so when it hits negative 90, that red set of doors that's currently open, those potassium channels, are going to close. And then we simply just repeat. Looking in the middle set of black sodium channels are going to open. That's going to cause sodium to rush on in. That's going to might make the charge spike and come all the way up to positive 50. And now we simply repeat what we said a moment ago. At positive 50, that middle black set of sodium channels are going to close, and the middle red set of potassium channels are going to open, and then potassium is going to exit, dropping the charge from positive 50 all the way down to negative 90. This is what an action potential, this is what an impulse is. It's the change from negative to positive to negative to positive. And so when the charge drops all the way down to negative 90, that middle set of red potassium channels is going to close. And look on the right. On the right, you have a set of black sodium channels that are going to open. And so at negative 90, the black sodium channels open, and the sodium rushes on into the cell, and that brings the charge all the way back up to positive 50. And same thing happens. At positive 50, the black sodium channels are going to close, the red potassium channels are going to open, and then the potassium will simply diffuse out of the cell. And that's how an impulse moves. That's how an action potential moves down the length of a cell. And so here's a real neat animation that shows the entire thing. Sodium comes in, brings the charge to positive. Potassium exits. Sodium comes in, brings the charge to positive. Potassium exits, and it goes back to normal. Sodium comes in, the charge is positive. Potassium exits, the charge goes back to negative. So here's a real neat animation at a faster speed right there. Well, you might be thinking eventually you're going to have a bunch of sodium on the inside that doesn't belong on the inside, that should be on the outside, and you're going to have a bunch of potassium on the outside that doesn't belong on the outside but should be on the inside. There has to be a way to clean up this mess. So who cleans up the mess? There's a pump called the sodium-potassium pump is going to clean up this mess. And how it's going to clean up the mess is through a type of cell transport called active transport with the use of ATP, adenosine triphosphate. And so what happens? Sodium is going to be pumped back out of the cell. Watching my animation. 1, 2, and even 3. 3 sodium ions at a time will go into the sodium-potassium pump, and with the help of ATP, when that third phosphate is broken off, heat is released. That heat causes the sodium-potassium pump to change shapes and therefore release the sodium to the outside of the cell. Well, what happens next is two potassiums will go into the sodium-potassium pump, and the pump will change shapes back to its original shape and then release the potassium back to the inside of the cell. So when does this happen? When does the sodium-potassium pump clean up the mess? Well, after the impulse is passed. Where is the sodium-potassium pump located? It's located in the cell membrane. And why does this mess need to be cleaned up? In order to restore the cell back to normal. This way another impulse can be sent in the near future. So if we look at this animation, I've now added the blue sodium-potassium pumps. I didn't add them before because it would have been too much clutter before. But we can see that 3 sodiums at a time are pumped out. I didn't show ATP, but I just wanted to get the idea of what the sodium-potassium pump is. And 2 potassiums at a time are pumped in. So you can see the cell is resetting itself. At the bottom one, 3 sodiums are released and 2 potassiums are brought back in. So there's 6 sodium-potassium pumps in my animation. Imagine if all 6 of them were going right now at the same time. The cell would be restored in a fairly quick amount of time. And so here we are back at our animation that we saw. Well, let's continue moving the impulse down the length of the axon. Eventually the impulse reaches the end. Well, let's zoom on in and take a closer look at this. When we do, here's what we see. We see a little gap in between the neuron on the left and the neuron on the right. That gap is called a synapse. So the definition of a synapse is the gap between 2 neurons. We have a neuron on the left, gap, and then neuron on the right. So as we saw a moment ago, sodium comes in, creates an impulse, and potassium exits. And so this process continues down the length. I'm just kind of reviewing from a moment ago. Sodium comes in, potassium comes out. Sodium comes in, potassium would come out. But now here we've come to our problem. That impulse has reached the end. It's reached what's called the terminal end. There's nowhere else it can go. That impulse cannot cross the synapse onto the next cell. So that's a problem. The solution are in those bubbles, those 3 circular shaped bubbles called vesicles. Vesicles hold proteins called neurotransmitters that are going to be sent from, in this case, the cell on the left to the cell on the right. So what happened, we just saw step number 1, an impulse has reached the end, what is called the axon terminal. Step 2, neurotransmitters are going to be released from the neuron on the left. Watch this. So that vesicle is holding 3 black dots. That vesicle will fuse with the actual cell membrane and begin to release those 3 black dots. Those 3 black dots are the neurotransmitter proteins. And you can see how one of them just landed on a receptor to the cell on the right. Once the cell on the right once a receptor on the cell on the right captures some neurotransmitters, that's going to trigger the opening of the first sodium channel. Notice how the black sodium channel just opened. Sodium rushes in, creates an impulse. Potassium exits. The same process happens. Sodium rushes in, creates an impulse, and potassium exits. Notice how the impulse started all over again to the cell on the right. Okay, so now I really am just reviewing because I just copy and pasted this from earlier. So now as we're following that new impulse, like we saw a moment ago, around -40 millivolts, the black sodium channels are going to open, sodium's going to rush on in, that's going to bring the charge all the way up to +50, that's the impulse. At +50, the black sodium channels will close, the red potassium channels will open, and potassium will exit the cell, dropping the charge all the way down to -90. At -90, that opened set of red potassium channels will close, and in the middle, that black set of sodium channels will open. Once that happens, sodium rushes in, brings the charge all the way up to +50. That's the impulse. At +50, that open set of black sodium channels are going to close, and in the middle, that red set of potassium channels are going to open, and that's going to cause potassium to exit, dropping the charge all the way down to -90 millivolts. At -90 millivolts, that open set of red potassium channels are going to close, and the next set of black sodium channels are going to open, causing sodium to diffuse on in, bringing the charge all the way up to +50 millivolts. You can see we're just repeating ourselves. At +50 millivolts, the black sodium channels close, the red potassium channels open, potassium exits, and the impulse again, or the internal charge again, drops all the way down to -90 millivolts. And the process continues. The process continues, of course, until the impulse reaches this location, the synapse again. So remind ourselves that the impulse is going to reach the terminal end. So sodium comes in, there's the impulse, potassium exits. That's going to cause the next sodium channel to open. Sodium comes in, creates an impulse, and then the potassium will exit. That's going to cause the next sodium channel to open. Sodium comes on in, creates an impulse. And so the impulse has now reached the terminal end. So the neurotransmitters are going to be released across that gap, across that synapse. So there goes a vesicle. It fuses with the cell membrane and slowly is taken into the cell membrane and releases the black dots, the neurotransmitters. Notice how one of the neurotransmitters landed on a receptor to the cell on the right. That's going to trigger the opening of a sodium channel, which will then allow sodium to diffuse on in, create an impulse, and then potassium will exit. That's going to cause the next sodium channel to open, sodium comes on in, creates an impulse, and then the potassium will exit. So you can see we really are just repeating ourselves. And you know, when we look at this, let's say you wanted to do something like kick a soccer ball. Let's say you're looking at a soccer ball, you're staring at it, your brain you get the idea to kick. So your brain is going to tell muscles in your foot to reach back and kick. Well, how does that work? Watch this. Your brain is going to send an electrical impulse down cell number 1. At the end of cell number 1, neurotransmitters will be released. Cell number 2 will create a new impulse. The impulse will travel down cell number 2. At the end of cell number 2, that impulse will trigger the release of more neurotransmitters. Cell number 3 will catch the neurotransmitters and create its own impulse. The impulse will travel down cell number 3. Eventually, the impulse reaches the end of cell number 3, and the impulse triggers the releasing of more neurotransmitters. Cell number 4 catches the neurotransmitters, creates its own impulse. The impulse travels down the length of cell number 4, and eventually the impulse gets to the bottom of cell number 4. that impulse will trigger the release of neurotransmitters, which land on cell number 5. Cell number 5 creates its own impulse. The impulse moves down the length of the neuron. Ultimately, the impulse is going to reach the muscle cells in your feet. Remember, if you're trying to kick a soccer ball, you're sending a signal down to your foot. So eventually, here's the impulse. It's finally reached the muscles of your foot. That impulse will actually cause muscle cells to move. That's what causes you to kick or to pick up something or to just get out of bed. If you want to just get out of bed, your muscles are reacting to electrical signals sent from your brain. By the way, remember the name of the neuron in the picture? We talked about it earlier. It's called a motor neuron. So there you have it. I hope you understand why I chose my animation now of people doing the wave. And action potential is kind of like people doing the wave. If you're in my biology class, pause this video. Bring me your answers on a separate sheet of paper. I'd love to check them for accuracy. So pause the video, try to answer these. Good luck.